Again, I'm going to go through the main things about your this evolution for today. So just to remind you all, whenever they test an evolution, right, then usually for some of the topics or some of the questions, there's usually a template that we need to follow. And you will always see the keywords are always the same. So one of them is like your natural selection as well as your, again, speciation, all right, like your macroevolution. But without further ado, let's talk about your this 4 types. So the 4 type questions they like test is, first one is your Darwin's theory of evolution. The moment they test you on Darwin's theory or anything about your this fossil data, this is the first type of questions they like to test. Second type of question is likely to test your this microevolution. Most of the time it's going to be your natural selection, and then there is always that key standard phrase or answers or a template that we tend to write for your this microevolution. The third one is always your this macroevolution. The moment we have macroevolution means there's speciation. Sometimes there are 2 types. They ask, like, for example, how does the evolution of this particular 2 species comes about? Okay, there will be an additional phrase that we have to add on from the natural selection type of questions. And then later, the 4th one is always the classifications. All right, so these are the 4 kinds, but let's go through the main things they like to test. So first thing is that whenever we want to talk about your this evolution, they always like a split between micro and macro. This is not found in your notes, so if you want, oh yeah, Myra, do you have? So again, if you actually need, you actually can write on your this first page of your notes, okay? Microevolution, please take note. Come, let's try. Can you tell me what is the definition of microevolution, Xianfei? Elise? It's always? So this is always changes in allele frequency. Is it within a species or again beyond a species? So it's within. Okay, a species. Again, haven't finished, is what? Within a species of a population. Now this is always the definition of microevolution. Definition of macro is always what going to be? Evolutionary. So it's again, please take note. Definition of macroevolution is evolutionary changes beyond a single species. So evolutionary changes beyond a species. This is a definition of my macroevolution. So in the moment you have macro means you have the formation of 2 or more species. So one main things, right, that you see they have both in common is that, if I want to talk about common now, they are all both driven by the same agents of your this evolution, which is your natural selection, all right? But the only thing they differ is number 1, okay, the scale of phenotypic changes, and number 2, your this different time scale. So again, sometimes a question will ask you, what is actually the relationship or link between a micro and macro evolution? I want you to take note, macroevolution, the link between them is that macroevolution is actually the consequence of effects of accumulated microevolution events. That means you have many, many rounds of microevolution taking place over many generations, which will result in what we call the speciation here, okay? So the link is, this is actually the consequence, so I'm just talking about this. So macro, all right, is the consequence of the effects of accumulated microevolution events. So when I say this is meaning I have many rounds of microevolution taking place, okay, that results eventually in macroevolution, okay? Now, so what is actually the main difference? If I want to draw as a table, so you can see the main difference between micro and macro. This is by right the definition between micro, this is the definition between macro. Remember I say the first difference between micro and macro is actually the scale of phenotypic changes. So how is the scale of phenotypic changes going to differ? This is helping you Because in exams, sometimes they will ask you to identify, is it a microevolution event or is it a macroevolution event? So you always look out for these two evidence here. Okay, so one, like I already said, is the scale of the phenotypic changes. The other one is that they have also the time scale. Time scale is very straightforward. Idea is that you should know already when will this microevolution be involved. If they already tell you in the question now, this is in a shorter period of time. By evolution terms, can you tell me, Megan, what does shorter period of time actually mean? Lesser than? Yes, the moment you see, they say, right, okay, this one actually occurs within or lesser than 1 million years. You can straight away tell yourself that this is actually a microevolution event. Understand? So again, let me just write down. So changes occurs over a shorter period of time. This is the most direct way of identifying whether is it a microevolutionary event or a macroevolution event. I repeat again, in terms of evolution, time, shorter period of time means that it's lesser than 1 million years. So if I say the changes occur a longer period of time, by evolutionary terms, this means that it's more than 1 million years. And I can guarantee you it's very obvious. They will tell you things like, okay, this one occurs 2 million years, then you have the evolution of maybe a Kia or actually a kiwi. Okay, so it's like two different species of birds. Speciation has already taken place. Alright? Second thing is, you look at the scale of phenotypic changes. This is always categorized by very smaller scale, of changes in body plans or way of life. Alright? This is the largest scale here. So as long as you see, let's say there's just deviation in, let's say, the body size, or maybe, for example, deviation in color, that is straight away microevolution. Okay, whereas when you have again much larger scale in changes, so they are such that they are no longer able to interbreed, that is already a macroevolution. Okay, so again, let me just write so you can see. Whereas this one, know the main difference between this micro and macro. Now, again, that's why know the link between micro, know the link between macro, they are all linked to each other. Now, again, we also see in your first page of your notes, second column, you always see how population is always the smallest unit of evolution. So the question is, why will evolution never occur within the individual level? Satu, dua, tiga. First one, can you tell me? How come evolution is impossible at the individual level? How come must be within a population? There are three, So again, for those who just receive your notes, you can actually write at the side, which is at the second column. First one is what, dear? What did I emphasize? We're just going through the basic.
Because individual got genetic makeup.
Same what? Same genetic makeup. So again, let's ask, what's the definition of evolution? Evolution is what? Changes in allele frequency, right? So in other words, to actually change the allele frequency, first of all, you must have individuals that must have interbreeding. Then you can pass down these traits to subsequent generation, right? Question, can an individual interbreed with alone? No. So the first one, again, you can write is that why population, or why is it impossible for evolution, to occur at individual level? Or you can actually tell me Again, why population is the smallest unit? Because number one is that population is a group of interbreeding individuals belonging to a particular species, and they kind of share the same geographical area. Okay, so again, individuals cannot interbreed. So again, that is actually, all right, one reason. Myra, what's the second reason? Think about this. Usually, what is the changes in allele frequency? What's the main mechanism in the changes of allele frequency that especially both micro and macro will share in common? It's all driven by what agents of evolution? Natural? So, natural what? Like always take place. Nature selects. So it, no, natural selection. Can you tell me right now, what's the prerequisite for natural selection? Prerequisite for natural selection is what?
Variation.
Again, we always must have variation, but what kind of variation? Phenotypic variation or genotypic variation? Because there's a difference, right? Phenotypic or genotypic? It's actually phenotypic variation. So will I have phenotypic variation within a population? Yes, right. Can an individual have every single variation of the gene pool? No. So again, the second thing is, again, only a population has phenotypic variation in the population for natural selection mechanisms to operate on. So I repeat, only a population has phenotypic variation for natural selection mechanisms to operate on. Individuals are merely selected for or selected against. Wani, this is not in the notes, especially the link. Okay, so take note. Then the next one is what? Only a population gives the definition of evolution. Only a population has what? What's the definition of evolution, Kalantia?
Changes in frequency within the population.
So changes in allele frequency, right? Agree? Can I see changes in the allele frequency within an individual? No. So we must have the gene pool, then we see the changes in allele frequency here. So the third one is only a population will have changes in allele frequency within the gene pool. So do take note, okay? How evolution, same thing. Smallest unit is always your this population. Clear? Okay, so this is one simple question they always like to ask. Microevolution, macro. Sometimes I also see what is the link between micro and macro, and sometimes they will say, give me evidence that why this is both a micro and macro evolution event. So that's why I'm going through this. Second type of question they like to ask, and you will see this in the question itself. That means the keyword in the question, which is called Darwin's theory of evolution or theory of evolution. The moment you see the word theory of evolution, they always like to ask the same thing. How does this, again, support the theory of evolution? So if you ever see, I said before, if you ever see the keyword theory of evolution in the question, what are always the two key phrases, okay, that we always must input in my answer here for theory of evolution?
Descent from common ancestor.
So descent, agree. Then what's the next one?
From a common ancestor.
Common ancestor, Yahoo, is with
Modification.
yes. Whether you want to put them together, you always must have these two together. Okay? Why? Why am I separating this? For your theory of evolution, you always have to give me two different evidence that prove descent. And then how come they have modification in their homologous structure. So I mentioned before, if you want to say descent, this is always descent, okay? Because it's all derived, okay, from a common ancestor. And this is actually how they inherit the homologous trait. Modifications, you must tell me why is it modified, okay? Due to different
Selection pressure.
yes, selection pressures for different functions. Okay? So again, let's ask starting with you, Wani. can you tell me what's the first evidence we always search for in your this question, right? To prove that they are descent that is derived from a common ancestor. First one is what? What are the evidence we always look out for?
If they come from the same location.
No, what's the keyword we look out for? Side H. We always look out for
Homologous structures.
yes. I must see, you must tell me, oh, they have or share the same homologous structure, and you tell me what. Sometimes in the question, right, they actually show you what is the structure that is homologous from each other. Please take note, homologous means that, again, there are similarities that are derived from a common ancestor. For example, your bone structure, your four limbs, okay, for humans, cats, bats, whales, they all have the same bone structure for the four limbs, right? Because the opposite is called analogous structure. Please take note now, analogous structure, side A, analogous structure, it means similarities, but those similarities, right, is due to convergent evolution. They were not from a common ancestor, okay? For example, your bat wing, your bird wing, your butterfly wings, all these are all for flight, right? But they all are because derived from different ancestors, but they share very similar selection pressures. Understand? Okay? But I say, usually this is the number one factor we look out for. Let's say, right, in the event I cannot find any homologous structures as evidence, what do we look out for the second evidence to prove they must be descent from a common ancestor?
Amino acid sequence.
Homologous structure, I just want to point out, it can be both, right? Again, your this morphological as well as genetic. This is what I mean by your this homologous structures. That means they share homologous DNA sequences, amino acid sequence. That's already part of your homologous structure. Okay? But let's say cannot, then we look at what for? Look out for what?
Close geographical proximity.
Yeah. So I notice how I never start this because the number one, okay, or the main evidence we always search out for in exam is your homologous structure. Clear? Now, modification. So let's try. I already said you always must write the keyword, which is what? Not only is derived from a common ancestor, we always must tell me. With modification, so how come the homologous structure look very different from each other? I'm underlining because they are keywords. So how come they look very different from each other? Because straight away, let's say I gave you some questions. Sometimes you can see it's a 5-mark question, right? The moment you see so many 5-mark question, how come they are modified? What's the key thing what's the agents of evolution that cause that modifications of the homologous structure? Due to natural? Sorry?
Variation.
Natural selection. Natural selection is the main mechanism, the main agent of evolution that will always cause this modification that will lead to differential reproductive success. You understand? So in the event, okay, let's say, right, this is just merely 3 marks. All you need to write is what? This, let's say, they say theory of evolution. How does this prove theory of evolution? 3 marks. You just tell me this is descent. Tell me what are the evidence, okay, that show that they are homologous structures, okay? So it means that, okay, they are similar, so it means they must have derived from a common ancestor, alright? And then again, the other 2 marks should be about the modification. At least, let's say if just 3 marks, then you must just write, oh, because they are modified due to different functions based on the different selection pressure due to natural selection mechanisms that operate on it. However, if this 5 marks, what must I always do? Write down every single thing to describe natural selection, right? 1, 2, 3, 4, 5. Sure, straight away. Number 1. What is always natural selection is micro or macro, by the way? Micro, right? So can you tell me, what are always the same thing? I'm just writing keywords. There is always a template for natural selection. You can see you do a lot of different kind of evolution questions, right? But the moment you are trying to describe natural selection, different schools, different questions, but the key phrase are always similar, right? What's the first one that we always need to describe for natural selection? There is?
Different selection pressure.
你的头 Heaven, what's the mechanism for your this natural selection to operate on? What's prerequisite? In order for natural selection, you always must have this.
Variation.
Yes, cannot just stop there. I'm just writing key phrase. Variation due to Yes, okay, due to mutation. Okay, that usually you will just elaborate that arises whatever favorable allele, but if they never specify, then you just write this. Next one. Yes, you must, I'm just writing selection pressure, but in exam, let's ask if they give you what is the selection pressure, must you specify? You must specify. What if they never give you? Then how? Then what must you say? Must you still say selection pressure? So they have different selection pressure. You still must write what this key phrase into your answer. I repeat, all this key phrase is you memorize keywords so you can fit into your answer appropriately. Make sure whenever you write your answers, right, it's always towards the context of the question. And then after that, can you tell me, Myra, after the selection pressure, then what? What's the next key phrase? You tell me what are the traits that are selected?
Selected.
4, because they are selectively advantageous. Again, you tell me what are the selective advantageous traits, or you tell me either this, or you tell me what traits are selected for. Again, after that, what must we always mention? Calanthea. Why? So what's the next key phrase? Any idea? All these traits they are selected for, that means the individuals are better, able to, because they are able to, they have selected for survive and yes, correct. So survive, okay, and reproduce, so higher, sorry about my English, and have higher reproductive success. Alright, to pass down traits. Don't just stop there. Always tell me, pass down whatever favorable alleles to subsequent generations. I'm writing key phrase. Any question? This is again micro or macro.
Micro.
So again, must I mention over time or over many generations?
Over time.
Over time. Must I specify what speciation occurs? No, because there is no speciation first, but if in this case, then must have one, right? Agree? So over time, at least tell me natural selection. So over time, or over in this case, this particular question will be over many generations because they are different species now, right?
Okay.
So please take note, the moment you see is speciation, you always must write over many generations, alright? There will be changes,
In every frequency.
yes. Now, if let's say, only if it is like speciation has occurred, then you must tell me, okay, that eventually it will evolve to form a new species again, or you can say speciation has occurred. Usually, you can specify, but sometimes you just say speciation of this two species form. Okay? Alright? So I'm just writing the keywords associated with natural selection now. I guarantee you, different schools, right, you always see this similar key phrase that appear over and over again, and they particularly bold and underline in your answer sheet, right? Agree? Clear? Now, so this is actually the first type of questions they like to test, theory of evolution, that I want to emphasize. The next thing is, you all know all the evidence of Darwin's theory of evolution, right? I'm not going to talk about the comparative anatomy, the embryology, but what I want to talk about is this fossil data only on transitional fossils. You all know what transitional fossils? So transitional fossils, why I want to talk about this, because I've seen in questions before, they relate transitional fossils with theory of evolution. So the question will always be, why transitional fossils can provide strong evidence for Darwin's theory of evolution? Always tell yourself this is based on one major thing. Because what are transitional fossils? Can you help me find some space to actually write in your notes? Okay. So find something, your heading, right? Your heading can be like, again, why transitional fossils? Okay. Provide evidence for Darwin's theory of evolution, or provide evidence for theory of evolution. First one, you can write, because again, is one main reason. The main reason is because transitional fossils reveal intermediate links between an ancestral group and its descendants. I repeat, transitional fossils reveal the intermediate link between an ancestral group and its descendants. So we need to elaborate why is this crucially important for this theory of evolution. Again, this is especially important now, so you can elaborate here. Especially important when ancestral group and its descendants are highly different. Another importance can be, so I repeat, this is important because especially when ancestral group and its descendants are highly different. For example, lizards and snakes. So what is a transitional fossil? Transitional fossil can be a snake, right? If you manage to find a snake with hind legs, right, or legs, right? Agree? A long-like feature. Or second reason, okay, why is it especially important is that when there's accumulation of modifications that are not apparent. Okay, so either when the ancestral group and its descendant groups, right, are highly different from each other, so you want to find a link, so a transitional fossil is crucial. The second one is that, let's say we have accumulation of modification because we have many rounds of natural selection taking place, okay, but that accumulation of modifications are not apparent between these two species that we are trying to compare, right? Because one thing about theory of evolution is that we always must prove they are descent with modification. So now you understand? Okay, so this is why I want to go through this. So this is the first type of questions they like to test. I'm trying to explore the many type of questions. Second type of question they like to test is your microevolution. For those who have your notes, can I borrow them? For those who have notes, you can actually flip. The heading will be called Process of Evolution. Okay? Now, for this process of evolution, okay, because we need to show how we can have changes in allele frequency all the time, right? Now, but one thing I want to point out is, for this one, I have the 5, okay, process of evolution. We just basically discussed the 5 ways that we can actually change, all right? This allele frequency is always obviously mutation. Okay? Now, one thing we already learned from your mutation chapter, we have those that are gene mutation and those of chromosomal mutation. Idea is that all this is going to increase this gene, all right? Because the same agent of evolution I emphasize time and time again is your natural selection. But let's ask, for this mutation, let's do a recap. Whenever we have mutation, we are looking for those that can have missense mutation that not only change this genotype but must change your phenotype. Because remember, environmental factors, they are selecting for phenotypes that are advantageous, right? But let's do a recap. Gene mutation, we have 3 types of gene mutation, right? Silent, deletion, insertion. Out of this, which are the ones that actually can give rise, Chloe, to this new phenotypic changes? You know what I'm asking, right? We have the 3 types, right? 1, 2, 3, silent. Sorry, my bad. Let me repeat. It's your substitution, deletion, insertion, mutation, right? Which ones are the ones that will always give about, let's ask a simple one first, missense mutation. Because missense mutation has a higher likelihood of bringing about phenotypic changes, right? Which one? Substitution. You agree with her? Substitution, deletion, or insertion? Deletion and insertion. Because deletion and insertion can bring about what we call, what's that thing we call? Frameshift mutation. Substitution mutation is less likely, right? Agree? Even though they can bring about, they only usually can bring about only one or few amino acid sequence changes. But why frameshift mutation is so crucial? Can you tell me? So why frameshift mutation can bring about more extensive, okay, phenotypic changes as compared to this substitution mutation? Sorry? No, frameshift mutation. Remember there's a key phrase that I mentioned in class? This is the key phrase, always tagged to frameshift mutation. So obviously this frameshift is a deletion of 1 or 2 nucleotides, right? Agree? So what is the keyword? That means nucleotides downstream are, keyword is what? Oi, I say you are memorizing key phrase to fit into your answers appropriately. So nucleotides downstream are, from the mutation are, improperly grouped into codons. So I repeat, nucleotides downstream are improperly grouped into codons. What's the next key phrase? Which will result in, keyword is what called extensive missense mutation. So when you have extensive missense mutation, wouldn't that greatly alter the 3D conformation of your protein? Agree? So that's why, again, we learned that eventually you can bring about greater phenotypic changes, right? But again, why substitution mutation is less likely to bring about genetic phenotypic changes, my bad. Phenotypic changes, can you tell me? 1, 2, 3. First one. What if I change this nucleotides within the gene of my DNA? How come there is sometimes less or lower chance for missense mutation to occur? So, you bring about what kind of mutation? Silent mutation. So what is again, why genetic code degenerate can bring about silent mutation? Oi, give me the way you write for exam. Because a codon, amino acid can be coded by, so hence, due to, yes, due to wobble-base effect. Okay, so that's why you can bring about silent mutation. This amino acid sequence or your conformation of protein will remain unchanged. Next one, Mara. What else can also explain why this substitution mutation is less likelihood of bringing about missense mutation and phenotypic changes? It's because maybe the amino acid sometimes maybe got missense, right? But why usually may not change phenotypically? That means your mutation can, I accept. Because do you agree introns are part of your gene? So maybe I have actually changed this nucleotide sequence within the intron regions, right? But what happens to introns during post-transcription? They are excised, and then again, you are only ligating my exons, right? So your amino acid sequence, your conformation of proteins will remain unchanged. Next one, Kalanthia. What if it's a missense mutation? Missense mutation means you change your amino acid. But sometimes how come change in amino acid, your protein conformation remain unchanged? Remember,
on the structure.
So what's the keyword? Not within the critical of the protein, right? So what kind of amino acid is that's most likely going to be? Non-essential amino acid. Maybe the missense mutation is on a non-essential amino acid, which is not found within the critical region of the protein. I gave you an example. I said the enzyme, right? Active site is the critical region. But let's say you change the amino acid is outside of the active site. It can still bind to substrate one. So function remain unchanged. Most likely your phenotype remain unchanged. What is the last one? You maybe replace the amino acid, but the amino acid has similar R groups. So when you have similar R groups, you can form very similar R group interactions. So the conformation of proteins may remain unchanged. Understand? But obviously, when we talk about mutation, relevant to evolution terms that can increase GV, we are talking about your gene and this chromosomal mutation that will change this protein structure. Alright? So, idea is, hopefully you know, chromosomal is both your structure and your number, right? Doesn't mean your chromosomal mutation does not contribute to this genetic variation. It still does. Because if I have, let's say I do here, if I have chromosomal mutation in the numbers due to non-disjunction, okay, wouldn't that already have unequal distribution of chromosomes? So if you have different number of chromosomes, will you already deviate from the diploid number of the parent species? So that will give rise to this increase in GV1, right? Agree? Not only that, I also can have chromosomal structural mutation. Example, what if it's, let's say, a deletion mutation? or again translocation mutation. So same thing, you may actually change, all right, your this expression of genes, or even let's say your this formation of your this gametes. So same thing, all this is supposed to give rise to your this mutation here. Gene mutation, you should know already, can give rise to what? New alleles. And when you introduce new alleles, are you changing the gene pool? Yes, you're changing the allele frequency within a gene pool already. So that's why both can increase GV. Now, I also mentioned you can see gene flow in your notes. Gene flow will also increase GV, all right? So take note, because you are introducing new alleles from one population to another. So this is what gene flow is about, all right? I'll just write here, you can increase GV. All right, so next one, we also have your this genetic drift. You are expected in syllabus to recognize, okay, genetic drift. This will definitely decrease GV, all right? And the two types of genetic drift are your bottleneck event and Midas founder's effect. I said there are two different scenarios, but they share, again, one thing in common. Because one thing you should learn, that the daughter population from these two events will no longer be representative of the parental population. Example is, by right, you must know what's the definition of bottleneck event. Megan, what's the definition of bottleneck event? Drastic decrease in population size due to what two things? Natural or mass predation. Understand? So again, or mass predation or your natural disasters will cause drastic decrease. Do you see in your notes? I have there. Okay, so highlight the drastic decrease. And I show you bottleneck, right? Do you see the diagram for a bottleneck? So example is, in that bottle, do you see originally the balls represent, the color of balls represents the different alleles. So do you see after the bottleneck event, originally the blue balls, white balls are about equal proportion, and you have some yellow balls, alright? Agree? But if I have natural disaster taking place, do you agree the blue balls are now overrepresented? The white balls are underrepresented. And what happens to the yellow balls? Eliminated. So that's why no longer representative. Founder's effect is also another one. Founder's effect is when you have a small migrating group of individuals that migrate to an empty ecological niche. So from this small migrating individuals, by chance, usually the proportion, right, of alleles, okay, that you migrate out, all right, through this gene flow is again no longer representative of the parental population. So that's why, same thing, they all involve a small population and they all involve that drastic decrease that eventually will not represent the allele frequency of the parental population. Understand? Next one, artificial selection, but I want to focus on is inbreeding. Especially, you should know, bottleneck event, founder's effect, have a higher chance of inbreeding. Inbreeding will drastically decrease this genetic variation. Okay? Because we always say inbreeding will depress the fitness of the organism. In fitness, in terms of fitness for evolutionary terms, it means that less able to interbreed to form viable fertile offspring. Alright? So inbreeding, right, would definitely decrease GV because of higher frequency of sibling mating. Alright? Next one is the more important one. So I want to focus on this because no matter what, we are always focused on natural selection. And you are expected in syllabus to recognize the three types of natural selection because sometimes in exams they will ask you to identify. But let's go through this. For your natural selection, just now we already learned the key phrases that are related to your natural selection process. Later I'll talk about why they decrease GV, but let's learn how to identify the three types of natural selection. The first type of natural selection is what, Myra? Can you tell me? What's the first type of natural selection? Because we have three types. And I said, please learn an example so you know how to recognize in questions if they ever test you. So what's the first type of natural selection? Sorry, D? Oh, wait. You must know the three, because in exam sometimes they ask you to identify. It's called? Okay, you flip in your notes. You see the first one. What do you see there? In the red box, red table. Directional selection. Now, directional selection is about what? What am I selecting for? Please take note, directional selection, you're always selecting a 4 against one extreme. So when you select a 4 against one extreme, okay, it causes the shift, alright, towards the other extreme. I'm going to give you an example. Notice how I purposely draw bell-shaped curve. An example, I said already. Please learn an example so you know how to recognize in exam, is it directional, stabilizing, or your disruptive selection. So, example will be what? If you see in your notes, I have, let's say, pesticide-resistant insects. So, for example, if let's say the selection pressure, alright, is the mass usage of insecticides, alright, or pesticides, wouldn't that be going to select for those that have pesticide resistance? Okay, so those without pesticide resistance is selected against. Understand? So that's why do you see it causes a shift? This is called directional selection. Valentia, what's the second type of selection? Stabilizing selection, select for what? Select for what? Select for the intermediate, but again, it selects against the both extremes. So stabilizing selection, as you can see, bring about phenotypic stability. An example for stabilizing selection, I said, please learn an example so you know how to associate. Alright? So the example I give is the gall-making fly. So what is the gall-making fly? Just to give you context, I'll be sending this summary slide again for those who don't have. So the gall-making fly, this is actually the gall. The larvae is actually within. So we have large galls, that means the diameter. We also have intermediate sized galls and small galls. The small galls, again, are very vulnerable against parasites because parasites can easily penetrate through and feed on the larvae inside. The large galls, okay, in diameter, they actually are very vulnerable by small birds. Small birds can easily reach these galls and feed on the larvae inside. But the intermediate sized galls are selected for. Why? Because they are too large for the parasites, too small for the small birds. So this is a type of stabilizing selection. You are expected in syllabus to know they select for the intermediate. Alright? Next one, we also have what kind of selection, Wani? Disruptive selection. Disruptive selection select against what?
The intermediate.
And you select for your both extreme. So again, when I select against the intermediate, you realize they will always bring about what we call bimodal selection. Example I gave was what? Your beaks of the African finches. Finches are birds. So, what about the African finches? So I gave this by right. Again, we have African finches with beaks of large beaks, small beaks, and intermediate sized beaks. Those that are small beaks are very well adapted in cracking the soft seeds. Those are large beaks are very well adapted in cracking the hard seeds. But the intermediate are not well adapted in cracking both types of seeds. They will be selected against. Okay? So that's why do you see this actually brings about the largest evolutionary changes? Please learn how to recognize the 3 types because I said already in exam, they will always ask. But let's ask this question first. First of all, again, this natural selection, do you think, you notice how I always say it decreased GV, right? But let's understand how come each of them will definitely decrease GV, alright, before I go through. So for this case, we know that directional selection. Do you see how come this decreased GV? You are selecting against, okay, those, again, pesticides, those insects that are not pesticide resistant, right? Wouldn't there be loss of alleles as a result? As long as loss of alleles from your gene pool, that is actually, again, decreasing your genetic variation. Alright? And then again, stabilizing selection, you also have loss, okay, of these genes from both extremes. You are thereby decreasing genetic variation. Same thing with your disruptive. As long as you cause loss of alleles, that is actually decreasing GV. So again, in exam, especially for essay, if they ever ask how natural selection decrease GV or decrease variation, please take note, you always must tell me the three things. What are the three things? First of all, you must always mention that it must be pre-existing variation. The pre-existing variation, right, undergoes what we have, this selection pressure. Alright? So that's why you're trying to show I already have variation, that's why the variation decreased. Second thing, you must tell me presence of a selection pressure from this environmental factors. Okay? They serve as selective agents that will select for individuals with variation. They allow them to outcompete, okay, others in the population. So that's why you need to talk about selection pressure. I repeat, you have to specify how the selection pressure, okay, will select for individuals within the variation and will cause them to outcompete others. Third one, this will definitely bring about what we call differential reproductive success. Which is just now, Kalantia, I made you chant is what? The individuals are better able to and have higher form viable fertile offspring, so you can pass down favorable to elaborate with effort to subsequent generations. I guarantee you the keywords are always the same. You are memorizing keywords so you can fit into your answers appropriately. Alright? Because you are trying to say that there's, again, those that have lower reproductive success, you are trying to tell me that they have less chance of passing down the alleles to this offspring. Alright? Such that eventually you must always mention how this will cause changes in allele frequency. Okay? You need to link it to the decrease in variation. So again, those that confer selective advantages to the individual will increase in the population or proportion, or those without the selective advantage traits or again their selected gains will actually decrease in population. So this will actually bring about that microevolution mechanism. Understand? Now, however, we know natural selection is always taking place. The prerequisite of natural selection will always occur. So if I say that natural selection always decrease GV, then how will the population always at least maintain GV or help to increase GV so that the natural selection mechanism can operate on continuously? Two things, right? We have mechanisms that increase GV and mechanisms that help preserve GV so that natural selection is always taking place. This is not found in your notes. I will summarize everything now. Into a table. Mechanisms that increase GV. And mechanisms that preserve GV. So mechanisms that actually increase GV, do take note, there's only three main kinds. First one is obviously your gene mutation. Okay? When we have gene mutation, how come will increase GV? It results in introduction of new alleles. Okay? And new characteristics. Such that we can increase gene pool for natural selection to work on. Okay, second one, chromosomal aberration. Okay, chromosomal aberration can result in new genotypes. New combinations of alleles. Idea is that not only you have new combination of alleles, remember I say you must result in phenotypic changes, right? So you must tell me how this will affect characteristics of the individuals. Okay? I'm gonna erase my space. What else? They also can increase GV. Jianfei, try. Actually got two more. What else can also increase GV that you see on the board?
Interbreeding.
Huh? What? Interbreeding doesn't increase GV. Interbreeding doesn't, is what? Gene flow. Idea is that the moment you introduce new alleles or new combination of alleles that gives rise to new combination of phenotypes, that's why they have different characteristics, that is increasing GV already. Understand? So the next one is gene flow. But when I say gene flow, I'm talking about the movement of gene. The movement of genes, okay, or alleles from one population to another. Due to migration, all right, and introducing new alleles. Into the population. So the moment you introduce new alleles, that is also increasing GV. There is also another one that increased GV. Any idea? What else also increased GV besides what I just mentioned? Me, yo, yeah. Okay, so let's do I will separate them. Meiosis, I write sexual reproduction. Get ready, 1, 2, 3. I want to see how meiosis increase GV. Gross meiosis. Okay, ready? First one, meiosis. Can you tell me what is the particular phase in meiosis that actually helps to increase GV? Prophase 1 that evolves. What's the way we write for exam? I want a full way. Crossing over between of, whole. How come that will increase GV? What's the way I ask you to write? Because this actually lead to new new combination of alleles. Next one. What is the next phase that increase GV? Of, whole. During So how come that will increase GV? What's the two things that increase GV from that process? Combinations of maternal and paternal chromosomes. What is the second thing? They also to lead to 2 to the power n possible combination of gametes. Next one, we are the last phase that also increase GV. What is it? Metaphase 2. What happens during metaphase 2? When they align in a single file, the sister chromatids are. Do you think the sister chromatids we are referring to is the identical sister chromatids or the non-identical sister chromatids? Non-identical. Sometimes the non-identical sister chromatids, right? Each of the maternal and paternal pair, okay, they face different. Yes. So what is the way we write for exam? The random orient of non-sister chromatids facing different poles of the cell during. Yes. How come that will increase GV? Because they can form different combinations of gametes. Understand? Random fertilization, you should know already. They also can bring about this offspring variation. Clear? Now, what else? We also know that, let's say, if you actually have mechanisms that increase GV, but I also have mechanisms that help me at least preserve GV, right? So let's go through this. First mechanism that help preserve GV, any idea, Columbia? Is called, like for example, usually heterozygote. Can I remove this recessive allele? Less likely, right? So what is that process called? Hetero? Your head, that is very different. Okay, heterozygous advantage is better able to survive for heterozygote, but this one you preserve this recessive is called heterozygote protection. There is a difference, class. Don't get confused. Heterozygote protection versus heterozygote advantage. Heterozygote protection, okay, involves when the dominant phenotype, okay, is favorable. So recessive alleles in this diploid organisms, they are kind of preserved in the heterozygous, right? Let me just write where recessive alleles. For example, if you want to have, let's say, pesticide resistant, maybe like the recessive, right, can still be preserved, alright? Because as long as you have one allele for pesticide resistant, you can develop the resistance, right? And it's still able to survive. But at the same time, you are preserving that recessive allele within the gene pool is not eliminated. Agree? Next one, we have your balancing selection. Your balancing selection is when you have your natural selection maintaining 2 or more alleles within the gene pool, right? We have 2 things that ensures balancing selection, right? What is the first? Can you tell me? Which is part of balancing selection. Hetero.
Hetero advantage.
Yes. Please take note, balancing selection means you are maintaining 2 or more alleles within the gene pool. This is part of balancing selection. I'll just bracket. Now, what is heterozygote advantage about? So, is that let me just write first. let me explain now. This is very different from heterozygote protection. Okay, heterozygote advantage is, alright, usually individuals who are heterozygous, alright, they have better fitness. When I say better fitness, in evolution terms, it always means higher reproductive success. Okay, so this is what better fitness means. I give you an example. Okay, sickle cell anemia and in a malaria-stricken country. We know that again, the heterozygotes for sickle cell have a higher chance of survival. Alright, why? Because homozygous for sickle cell, they are very prone to the sickle cell disease. They can die from, let's say, stroke, heart attack, etc. Right? Higher chance. Whereas again, those again for homozygous for the healthy hemoglobin are also very vulnerable to the malaria, which is a type of blood-borne disease. Agree? Because it can spread much more easily, affecting the individual. But the heterozygotes is, again, are that they have some, okay, they still have normal hemoglobin. So they will not be so vulnerable to your sickle cell disease. And again, half of your this red blood cells may actually be sickle cell. So that's why it does not spread the malaria, the blood-borne disease, as effectively. Alright? So that's why they are better able to survive. Clear? Next one, another type of balancing selection is what? Simfei? Frequency. I just write F. Frequency dependent selection. Please take note, this is also part of your balancing selection. Okay, so what does the frequency dependent selection describe? Usually it describes that if a particular phenotype, right, becomes too common, it will decline in numbers, alright? So the frequencies of different phenotypes kind of oscillate over time, okay? So So again, it's the left mouth and the right mouth scale eating fish was the example I have. If you can see your notes, it's on again, this picture your notes here. Okay, you see the diagram. Okay, so your left mouth and your right mouth scale eating fish. All right, you can see how they kind of just fluctuate within the mean. All right, so again, this is a type of your this frequency dependent selection. But we have more. What else can also preserve GB? Any idea, Chloe? I give you a hint. An example of hint is your human fingerprint. So it's referring to what? I erase this, I need space. So it's what here? Okay. You want to help her? So neutral mutations that bring about neutral variations, right? So what's neutral variations? Is genetic variations that have little or no impact to this reproductive success. So thus the natural selection does not affect this kind of variation. But again, why this one again can preserve GB? Because they still involve mutation. Okay, but the mutations do not result in changes in proteins, thus no changes in phenotypes can be one of them, or they do not have impact in the reproductive success. So there are many variations for this. One is your human fingerprint. So does it actually affect your reproductive success? No one. Every single individual have different fingerprint. Agreed? So involve mutations. Example can be your mutations in the non-coding region, alright? Or, okay, it It can also involve, alright, no impacts to the reproductive success. Example, sometimes this also can bring about is like the degeneracy of the genetic code. Maybe you have the same amino acid sequence, but you have different nucleotide sequence. And you can actually see this very obviously in very closely related species like chimpanzees and humans. But we are not the same. Okay, most of our these proteins made are actually about the same. But our nucleotide sequence are the one that differ us apart. Understand? So this actually preserves GB. So the moment you know that this question is actually testing you on your this evolution, right? Okay, that is microevolution. Please take note. Microevolution most of the time is always natural selection. But I want to highlight something. Natural selection, you know what are the key phrases associated to describing natural selection question, right? But sometimes the question wants you to specify what kind of natural selection taking place. Please know how to identify the 3 different types. This one I have here, the mechanisms that increase GV, mechanisms that preserve GV, is very rare, okay, they can come out in paper 2. But why I'm giving this is what if they come out in your paper 3. So please know, okay, clear? Next one, macro. The moment is macroevolution. The key phrase will always be as such. Macroevolution already shows you have, again, at least a speciation taking place. That means a formation of at least 2 species. Alright? Now, but sometimes in certain questions, they have both micro and macro evolution, and then you need to describe both within a single question, right? Like some students tell me, oh, when do I write the keywords for just natural selection only? When do I actually write the keyword for your this macro? Because sometimes, right, to showcase how you form a new species, wouldn't I still need to describe natural selection? Agree? So the idea is, the hint is, the moment you recognize this question is asking you to write about your natural selection, or you have to keyword, another key phrase they have is that how you cause evolution of this species, or how you form this new species, then that is also telling you need to write your macroevolution question. The keywords. So what are the things that I mentioned? Just now, right, first thing we said for natural selection, what's the first key phrase that we always write, Chloe? In the? But let's say you also have to write macroevolution, okay, kind of answers. I don't want you to just write variation within a population. You must say variation in the ancestral population. Okay, due to mutation. So if it's just like within the species or micro, then no need to write ancestral. Because everything comes from the ancestral species, right? So only when it's talking about speciation or when it's talking about macroevolution, then you must write the word ancestral. Understand? We still need to tell me, oh, different selection pressures. Okay, I'm not going to write down. Different selection pressures, okay. Changes in your this allele frequency over time. This one, we already know, we always have to write those key phrases in my answers, alright? But let's say, we have here, for your this macroevolution, just to form new species. Please take note, we still need to mention there is changes, okay, in allele frequencies, sorry, I forgot to write different changes in allele frequencies in different populations. Because you should know already, this is talking about separate population if you are talking about the formation of 2 or more species, right? So I must see not only just different changes in allele frequency in different populations. Then, what must you tell me? Keyword is, that is due to no gene flow. I'm writing keywords up. You have to make sure you know how to fit those keywords into your answers appropriately. Alright, may not be in sequential order, so you accumulate. Okay. Yes. Okay? Accumulate. Yes, sorry. Accumulate. Okay? Greater genetic differences. Okay? Such that, same thing. Tell me, usually you say greater genetic differences, obviously it's due to your natural selection, or again due to genetic drift, elaborate now, or due to mutations. Okay, so you must tell me how population no longer able to interbreed to form viable fertile offspring. Okay, so eventually you will say how. Sometimes you can actually specify, but you have to see carefully. If they give you a diagram that obviously has no genetic barriers, you don't tell me genetic geographical isolation and whatever. Okay, but you will definitely have reproductive isolation. So it will become increasingly geographically isolated. From each other. Okay? I'm just writing the keyword up. In the event, you sometimes have to write both natural selection and macroevolution together. Later we'll do some questions and you'll see why I say as such. Alright, but let's discuss. Because I said there is such thing as geographical isolation, there is such thing as your reproductive isolation. We need to learn the different isolation mechanism. Now, again, reproductive isolation, you are expected in syllabus to know there's four main reproductive isolation mechanism. Please learn how to recognize. First type of reproductive isolation is what? Any idea, Megan? Please know how to recognize. Oh, I forgot to mention. Last one, always end off, okay? What is the type of speciation? Alright? That has taken place. My bad, I forgot. Later you'll see the two types. Okay? So you need to usually reference to what kind. Then later you'll see there's two types, allopatric and then your sympatric speciation. Okay, now let's go through. We have behavioral isolation. Behavioral isolation, we should know is what, Megan? You too? Different what, sorry? So different mating rituals, what else? Not only just behavioral mating rituals, right? Can also be different phenotypes, such that the species do not recognize those phenotypes as their own member of the species or own species. Understand? So behavioral isolation is referring to either your mating rituals, can be the mating calls, okay, etc. Sometimes yours, if you were with me before, I show you like there's a video for some birds, they actually mate by performing and dancing, right? So it's different mating rituals, or you have different phenotypes where your species, only members of species of the same species will recognize them as their own. The second one is what, Myra? Start with T. Temporal. You know what's temporal isolation there? It's a type of reproductive. It's meaning different mating seasons, different times of mating. Okay? Sometimes, even though they may share the same geographical location, but they have different mating times that will not overlap with each other. Alright? Next one, any idea? Valentia? What are some reproductive isolation as well? Physical? This is what we mean. Oh, morphological. Wait, no, mechanical. Mechanical means what? Different gonads, different reproductive, okay, organs for mating. Alright? So mechanical is also another isolation. The other one is what? Post-zygotic isolation. That means, let's say your gametes fuse, they cannot form viable zygotes. Understand? Clear? Now, so this is your reproductive isolation. Geographical isolation, you should know already, they are just, again, going to be isolated within your area. But I said before, when you want to describe, we need to learn the two types of speciation, right? In your notes, speciation is actually found here where you see allopatric, but I want to go through sympatric first. Okay?
Sorry.
Okay, do you see here? And here is your this allopatric, behind is sympatric. I want to go through what's the difference because it's in your syllabus to know what's the difference between the two. For those who just received the notes, you can just write these two main key differences between them. First one, let's ask Moni, what is again the main difference between allopatric and sympatric? Because sometimes I say you need to specify, and this is part of your marking scheme. Is what?
One is repro isolation, one is geographical isolation.
Sure, but what is say? The idea is that this is the formation of new species due to presence of a physical barrier. Okay, whereas this one, formation of new species due to what, Wani?
Genetic difference.
Yes. Let's ask, there are again, since we are talking about the two main difference, physical barrier and genetic difference, what brings about the genetic difference for sympatric speciation? Xianfei? There are two mechanisms that bring about your genetic differences, not just any mutation, but it's what kind? Your? No, the two things that you see just now, alloploidy and autoploidy. My question is, what are actually the difference between the two?
Within the same species. Or no?
Which one? You asked me, I already answered you before. First one is what? Which one is within the same parental species? Which one? Auto? Sure? I may be gaslighting you, so make sure you know. So it's what? Autoploidy, right? Agree? So I want you to take note, it's actually found on this page of your notes, your first column. Okay? Do you see autoploidy? I want you to take note, autoploidy is within the same parental species. Sometimes it's due to this chromosomal duplication, or again, it can be due to this what do you call that? Non-disjunction. So as a result, right, you actually form a polyploid. Alright? So with this polyploid, do you see how it deviates from the diploid number of the parent species? So it means that this new species, do you think can interbreed with the parent species? Organism of the parent species? No. So this is the first one, autoploid. The next one, what do you see? Alloploidy is what? Xianfei? Is between two different species. Examples, okay, can be like these two different species, right, can be again two different species with different diploid number. When they actually fuse, they form a new zygote with actually the diploid number that differs from both parent species. Example will be like what? Okay, you're this liger or you're this tigon, or it's actually more prominent in plants. That's why you can see we have very unique kind of plants that can be polyploids. Example, your potato is a tetraploid, okay? Your this strawberry is this hexaploid. So they have polyploidy. Understand? So this is again one thing you need to take note of how we can bring about sympatric speciation. Another difference between allopatric and sympatric is what? The time period. Please take note, this is actually a very slow process. It must take place how long? Can you tell me before speciation occurs? Can you tell me, Chloe? How long must this occur before speciation can be prominent? Because speciation now you form new species. Yeah. Millions of years. Idea is that, let's say if this physical barrier is actually removed, okay, that means this two population is not able to interbreed. At most they form subspecies, but not new species. Please take note, subspecies is still within the same species. Like human, we have a lot of subspecies, right? What is our subspecies? Asian, European, Indian, etc. Right? Agree? Americans, blah blah blah. Right? Agree? So this is again what it means. Whereas the other one, please take note, it can occur in a shorter period of time. Sometimes it can be within a single generation. Okay? Because you just need this two. Clear? But then this is speciation. Then the question is, students always think, and then what if, how would I recognize that this is actually testing me on adaptive radiation then? Then what's the difference between adaptive radiation and speciation? What's the difference? They are actually, huh?
That one is
Many species. That means there's many rounds of speciation. Understand? It's just like macro is many accumulated microevolutionary events. So adaptive radiation is when you have many speciation, so it's a type of way to describe divergent evolution. So if you ever want to mention now about how do I recognize whether a question is testing me on adaptive, they always have a remote island or maybe a mainland, and then we have surrounding small little islands, and we found that surrounding small little islands, all these different species are closely related to each other, and that is already telling you that this is testing your adaptive radiation. But what's the key phrase we always tell and write for? Let's say if I want to include adaptive radiation in my answer, obviously we still need to come up all this, right? But the first thing when you want to write is that for adaptive radiation, you don't just tell me variation in an ancestral population due to mutation. You must say how, at this right below, is that all this were able to colonize, okay, new, different, empty niches. Empty ecological niches. Okay, then you specify the different niches have different selection pressure. Vomit out the same thing that you learned from this macro, okay, and microevolution. And then for adaptive radiation, usually you need to specify how are they going to be different from each other. So you need to specify this type of reproductive isolation mechanism that makes them actually no longer able to interbreed to form viable fertile offspring. Understand? Okay, so I'm just trying to showcase adaptive radiation. The way you phrase, right, they are all actually very similar to each other. You just need to know how to tweak and when to tweak. So I repeat, if it's testing your microevolution, just vomit out all the natural selection keywords, right? Depending on what the question is asking. Look at the question in the, look at the keywords in the question. Second is macro, just purely macro, just asking you what speciation. Add the natural selection, just tweak accordingly. Add those in purple, especially in red, and then you again specify the speciation. How will I know if it's adaptive? Either the question tell you again, explain how adaptive radiation occurred, or they show you a diagram where you have a remote island, or you again have one island, then surrounding got so many different species in close geographical proximity. Understand? Know when to write. Adaptive radiation is when you have many different species, and you can showcase more than 2 species already. Alright, so let's try some questions so you know what I'm talking about. Can you help me pass down there? Now, each one of you, I want you to do your respective questions for me to mark. Okay, before we try out some questions. Do you see we have question 2A and 2B? 2A and 2B, as well as 2C. Xianfei, Megan, and Kalantia, I want you to do question 2A, B, C. The rest of you, do you see we have question 3A and 3C? I want you to help me to do 3A and 3C. Please try these questions, and when you're done, let me mark. There will always be key phrase in the question, and then the things that I just emphasized in class. So far, I just went through the 3 types of questions they like to get.
Can I have my GC? Oh, sorry. Yes. Okay.
Questions can use this sign to ask. Question kena asal. For those who are doing question 2B, let's ask. Megan, wait, are you the one doing 2B? Can you now tell me, is this testing you on micro or testing you on macro? Macro, but not micro? Both. You know why? What's the keyword in the question? Examiners, right? I already said it so many times before. The way examiners choose to use words to phrase their question is deliberate. What is the keyword in the question we have here? It's like, do you see they already asked you speciation is taking place? Speciation is a part of outcomes of evolution, which is macro, right? Agree? But How come I know it's also talking about your this micro, which is mainly natural selection? Because of the word what? How. They ask how this speciation occurs. Speciation, we already learned, is from many accumulated microevolutionary events, which is your macroevolution. So, do you see this 5 marks? So they are asking how come I can form many different populations, okay, from this mice, right, species. So they must all come from an original ancestral species, right? Do you know how to write the associated keywords? Right? So the question is, I know there is no gene flow, right? What is the one that causes no gene flow, Kalantia? Or prevents gene flow? And by the way, don't ignore the paragraph in front. Examiners will never give you unnecessary information. So what did they say about the gene flow? Sorry, why no gene flow, Kalantia? Because of presence of steep mountains that serve as geographical barriers that prevent gene flow. But how come this population of this mice can it be scattered? How come I have 6 different populations even though it comes from an ancestral species? Due to? What are you talking about? I mean, like, how come at first they come from an ancestral species? Right? Why one ancestral population can be separated in 6 different kinds? Why?
Migration.
Of human migration and? So humans are the one that scattered them apart, right? So it always starts off, so that must be the idea. We start off from an original population and they were scattered from human migration and settlements. Examiners will never ever give you unnecessary information. Never. So every single information they gave you was with intent. Now, so later you scattered already, then later you say the steep mountains will provide the physical barrier, right? That prevents no gene flow, right? Different locations will now have different what, Megan? Because it's also testing on natural selection. Different locations will have different. And then, again, because those that will have selective advantage, must we specify what is the selective advantage or selection pressure? No need, right? Because we don't know, right? We just say different, right? Different selection pressures, such that different mice with different selective advantage will be selected for. We still write down the whole thing, right? Survive better, reproduce, pass down the favorable alleles to subsequent generation, which result in what? Eventually, what must we say, Kalantia? Once you have different reproductive success, then pass down the different favorable alleles to subsequent generation, because this is macro, right? Then what must we end up? Different? They have different? The one in purple. I don't want to just see over time there's changes in allele frequency, because there's different populations. Each different populations will have different changes in allele frequency, right? And then after that, you must tell me how they accumulate greater genetic difference such that they are no longer, because now you want to form new species, right? So what must we always write as well? To form? So they are no longer able to interbreed to form viable and fertile offsprings. However, must we specify how different they are? Huh? Like, how come we have greater genetic difference? What causes the greater genetic difference? In the bullet point, they have showcased something in the bullet point in front. Yeah. So you must specify the different mutations, right, occur differently, such that they develop different chromosome number or diploid number. That's why they can accumulate sufficient genetic differences. Understand? But I'm trying to showcase you see it's based on the template that I gave you. I don't call it template, that's a very misleading word. Based on the keywords that I always emphasize. So I repeat, girls, when I gave you keywords, it's not like, oh, you write down in sequential order. Not really necessary. You write down appropriately, but you're memorizing the keywords so you can fit into your answers appropriately. Understand? Okay? Next one, for those who are doing question 3C, they say with reference to the information provided. I say time and time and again, and chan again. Zoe, what does it mean when I say with reference to information provided? So I always need data, right? Agree? So, question, is this testing me on micro or macro? Sorry? Macro. And not just macro, okay? But must I also say micro? Yes. So what do you all realize? Most of the times, right, you look at the number of marks. If it's like so many marks, like 5 marks here, usually if you recognize it's macro, it's a bit of micro and macro. You just need to tweak your macro keywords accordingly. Which I already say what? Do you see the macro? You just need either tweak into ancestral population. And then if it's adaptive, hopefully you know what the additional things you need, right? And then you still need to write those in purple, right? Agree? You just need to tweak accordingly, what? But this time, you need to show me how is adaptive radiation. How come I know this is adaptive radiation? Obviously, because they use the word adaptive, right? Agree? And obviously, they say occur in a remote island. But sometimes if a question never write adaptive radiation in the question itself, then how come I know this is adaptive? The moment you see form more than 2 different species. In this case, they already say at least 7 different species of the Guanteloupe junco. Okay? So I know this is also testing me on adaptive radiation. Alright? So hopefully you know, must we specify the type of speciation, Myra, for adaptive radiation? Yes. Can you tell me right now, what kind of speciation will this be? Allopatric or sympatric? I already show you what's the difference between allopatric and sympatric. So, allopatric or sympatric? Yes. Because they have no geographical barrier. It's all within the same island, right? So this is sympatric. Make sure you know how to fit the sympatric speciation into your answer. Okay? Okay. Hopefully you know how to recognize. This is why I say please learn how to recognize. That's why I say evolution can score 1. Please do not leave evolution blank. And once you recognize, okay, the type of question they are testing you is quite typical. Once you're done, let me mark. While waiting, you can do the other question. Keyword, different species, relationships. What do you mean respond? Again, what do you mean respond? Because you're asking like why different species. You say different species respond to different kinds. I don't think that is actually, so they don't make sound. The other one make sound, right? So in opposed to your respond, right, would it be better if I say different species make different kinds of calls? Or the keyword is called, the calls are species specific. Learn this keyword. And that will count for the different species. And then the relationship, like how you investigate relationship. You telling me can use to identify different species will interbreed to form variable fertile offspring. Then I question, later you write, interbreed to form variable fertile offspring. This is within the same question. You also have to write the word no longer able to interbreed to form variable fertile offspring, right? Do you think examiners will ever want answers that is repetitive? So this is also a big indicator. What to write, what not to write. You understand? So again, first thing is, you say it's a behavioral isolation. Idea is that you do not show me the relationship between the different species. That means they are saying, right, how the different species are related to each other. So the idea is, you need to show me the relationship is, the greater the similarity in mating calls, the more closely related the different species are. Do you understand what the question is asking now? So that's why the keyword is relationship and then the different species. Clear? That's why I cannot give you any credit here. Alright? Next one, you should know already, adaptive radiation is the keyword. Variation in ancestral species due to spontaneous mutation. And then they colonize different ecological niche. Fine, I'll give you the one more here. So populations from different face different environments, have different selection pressures in the different niche. Very good. So again, you mentioned how individuals with favorable traits in the different selection. Next one, next time say different population can survive, interbreed, and pass on the favorable traits to subsequent generation. Very good. Next one, you never tell me over time there is different allele frequency changes in different population. So that's the missing thing that you should actually write up. Okay. And then you say due to sympatric speciation. Good. Gene flow is disrupted. So you tell me the behavioral isolation, but you never tell me an example what is the behavioral isolation because you didn't quote data. Hello, where's your quotation of data? Which is again, you underline. That means the eye color, plumage color, or the song variation. You need to quote the data. This is the data you have to quote. You understand? And then gene flow disrupted. Yes, good. So again, unable to interbreed to form viable fertile offspring. Let's see. Okay. I can give you still the full 5 marks. So what do you start to realize, Chloe? Do you see sometimes, right, you have, and most of the time to be honest, okay, is that the number of marks, okay, allocated is fewer than the number of points that we are looking for as examples. Okay, so can still score. Genetic drift. Isolating mechanism is just now this one.
Like the behavior temporal.
Geographical, behavioral, temporal, mechanical, and post-zygotic. So which one is this?
Post.
你的头啊 geographical.
Oh, geographical and therefore. I thought it was just.
Yeah, I said this one is only for this. But you also are geographical, ma. Right? And then hello, Patrick. That's why you write genetic drift, isn't it very off? Okay. Hey, right, isolating mechanism. So no, so it's only those. That's why I say. Okay. Sure. Due to spontaneous mutation. Next, you mentioned how Sunny Valley and Strip Mountain. Oh, different valley. They are living in the valley, separated by steep mountain. They are not geographical isolated by valleys there. The steep mountain, they already say separated by steep mountain. They never say separated by valleys. So please, there is different. You have this one, where's the no gene flow? Never right, I cannot give you the credit. So different selection pressure in a different population. Idea is those with more favorable traits will be selected for and able to survive, pass on to the next generation. Fine. The idea, you must just tell me, results in change in allele frequency in different populations. Different changes, not just changes. Different population and different type changes. So you mentioned there's accumulated greater genetic variation. What? Not due to leh. Okay, fine. Fine, I can accept. So no longer able to interbreed to found variable fertile offspring. 3, 4, 5. I can still give you the maximum mark. It's just a bit wonky. Now the keyword is outcome. So you are saying produce viable fertile offspring due to reproductive barriers. Huh? The thing is that why would they again ask for the same exact answer? The question will never expect you to have the same exact answer to any of the prior questions. Do you see we have the same exact answer as the one before? That makes no sense, right? So idea is that if I make these two different individuals together, okay, can you tell me they are of different species or the same species? Do you think they are of different species or same species?
Yeah.
Because they have different diploid number. So if they have different diploid number, will they form different gametes with different haploid number?
Yeah.
So what happens to the diploid number of the resulting offsprings when these two different individuals interbreed? It's different from both parents, right? So when you have different chromosome number, can you tell me, will the offspring, by the way, be again fertile or sterile? Which one? Sterile is almost like the donkey and horse, then you form the mule, or the tiger and the lion mate to form the liger. Are they actually considered species? Because they are all sterile, they cannot continue, okay, this production of offsprings. You understand? So you need to elaborate is based on that. So idea is because of that, due to the different diploid number or chromosome number. That's why, again, you need to further elaborate how come the offsprings are infertile. Because of the word explain, which is a command word. Understand? So way off. Who else? Thank you. Again, keyword, different species, relationships of these different species. The only way how different species are related are either closely related or distantly related, right? So this is what they are asking. Why you give me so much? Can I ask you something?
I don't know why.
In this case, right, you are writing how the sound of different calls. To me, no dear. When they ask you this, and this is 2 marks, usually 2 marks or less, it's very straightforward answers. So how come they are called different species? How are they known different species from the calls? Because the calls are different species have what? Different
Behavior.
calls. Different kinds of calls. Or the keyword is actually called what? The calls are species specific. This is the keyword. Understand? And then you never tell me anything about the relationship, so I cannot give you the credit here. Okay? You don't say me different calls are recorded. You don't just tell me this. They are not asking for different species. They are talking about how can you recognize the species from the calls. Because the calls are species specific. if again different species of songbirds, you usually make different kinds of calls. Then what's the relationship? Relationship is the greater the similarity in the calls, the more closely related the different species are. You understand? So learn how to recognize the keywords there. You underline, but doesn't seem that you recognize. Understand? They're not asking you how you investigate and then say that. They are saying how from the calls, then you can derive the relationship. Okay? Adaptive radiation. So you say variation in ancestral species due to popular mutation. So the idea is that different parts. Songbird's ability to occupy. No, you just say and they have the idea. Next time you tell me is that they have variation, and this variation in this one colonize different empty niches. That's all you need down. They colonize different empty niches. So be very straightforward. Variation in ancestral population, okay, colonize different empty niches, and they develop different mutations. Understand? This is how you actually phrase your answer. Clear? But I'll still give you the credit. So you guys say that different niches have different selection pressures. Fine. So in other words, different favorable characteristics are selected for. Those selected for better able to survive, to pass down alleles to subsequent generation. Very good. Next one, you must tell me over many generations there's change in allele frequency in different populations. Fine. The idea is different changes in allele frequency. Different changes in allele frequency in different populations. So you say that how eventually you must tell me what is the greater genetic mutations. What? You never tell me again. You just say accumulation, there is no gene flow. How come there is no gene flow? You never mention how come there's no gene flow. Why you say the different, you write the different niches resulting in greater accumulation of genetic differences causing no gene flow. What is the differences they have?
The allele changes.
No, the differences they have is the different eye, plumage color, song variations. So this is a behavior isolation. The behavior isolation is the one that prevents gene flow. I know I write no gene flow down there, but you need to specify what causes no gene flow. Is it the geographical barrier or is it the, again, reproductive barrier? So you never specify. I cannot give you the full mark. But actually I can because it's just 1 point. Okay, fine. So again, here, accumulation of greater, then this is the one, once you have no gene flow, then you allow the accumulation of greater genetic variation. Because you allow, when I say no gene flow, okay, how come no gene flow come first, then you accumulate? Because when there's no gene flow, each of the population evolve independently of each other and change different allele frequency from each other. Then that's why you can accumulate genetic differences. You understand? So again, at least you mentioned how they are no longer able to interbreed to form viable fertile offsprings. And this is a result from Fitzpatrick. 1, 2, 3, 4, 5. So you can see I can still give you the maximum 5 marks. Who else?
Can I ask for adaptive radiation? So you have to say about how it occupies like the different ecological niches.
Yeah.
Only for adaptive, like the normal one, I don't have to say. I'll just say different selection.
Or the moment you recognize it's an adaptive radiation questions, you have to. Okay? Variation in ancestral population due to variation. Inception. Variation in ancestral population due to what? Mutation. Okay? Which migrated into 6 different locations. Sure. Isolated by steep mountains. You need to specify, okay, when you actually scattered in this, you never specify how come they migrate into 6 different populations. It's due to human settlements. That's what human migration and settlements specify. It's not the thing now. Okay, so I cannot give you the credit there. You say isolated by steep mountains, you must tell me when you isolate by steep mountains, what does that prevent? Yes, that's the keyword you're missing. Understand? So different locations are different, so there is different selection pressures. I accept for this. So you can say how different selective advantage, with different selective. Hello, you need to learn how to fit into your answer. So if you have different locations, why not you say different valleys have different selection pressures? So that's why mice with alleles that confer selective advantage are better able to survive. So fine, I can still give you the credit. Okay, from natural selection. So that will result in different changes in allele frequency from different populations. No, you don't have accumulate greater genetic difference due to. No, the different diploid number, right, is the one that. Okay, fine, I can still give you the credit. Different here. Gene flow is disrupted is you must write is here. The one with the steep mountains. Understand? So they are unable to interbreed to form viable fertile offspring. Okay, so from your this isolation. Fine, I can give you the credit here. We do not know that whether is it allopatrica yet. But 1, 2, 3, 4, 5, I can still give you the maximum mark, just nice. The individuals with different diploid number of chromosomes, they interbreed to form different So why are they not fertile? Because the gametes, right? Can they actually, the question is you never explained to me why they will not be fertile. Reason why they will not be fertile is that the zygote form is very different diploid number from the parent species, right? So the question is, do you think I can have proper pairing of homologous chromosomes? If you have no pairing of homologous chromosomes, this offspring do you think can form even viable gametes in the first place? That's why they are non-fertile, not fertile. Okay, so you need to mention that. Who else? Thank you. Geographical, allopatric. So variation in ancestral population due to spontaneous mutation. Fine. And then human migration and presence of steep mountains. And then, Oh, okay. What are you writing? Very messy. Oh wait. Settlements of steep mountains. There will be steep mountains, then what? Miles ah. After the steep mountains is different valleys or the miles? Different valleys ah. And different valleys, they share different selection pressures. The idea is that the different mountains, right? You must tell me they are you never tell me how they are. The human migration and settlements will scatter these mice into 6 different populations. You need to mention that. So I cannot give you the credit for that. And then the missing is that the presence of steep mountain will therefore provide no gene flow. So that is also another keyword you're missing. But I'll give you the key answer that there are different selection pressures in different locations. Okay. So again, mouse with advantageous traits, selector 4, better able to survive, higher reproductive success. Okay, fine, I can accept. Hey, but why you never tell me to pass down? Okay, very messy are you? There will be different changes in allele frequency in different populations. Why must you say due to? There's no gene flow. Accumulation and genetic differences from the different chromosome number, no longer able to interbreed to form viable fertile offspring. The question is, you never tell me the reproductive success to pass on the alleles to subsequent generations. Hey, you're not writing the key things. But I can give you 1, 2, 3. Oh, I can still give you 5 marks. So you can see the beauty of evolution. Sometimes the number of marks is fewer than the number of points we are looking out for. Okay, so always elaborate. Result formation of non-fertile individuals. You never tell me how come non-fertile. I only can give you 1 mark, but at least you can identify they have different diploid number. Next one. Keyword in the question is called different species from your calls. And then the second keyword is relationship. Different species have different behaviors. What do you mean different behaviors? Or different mating calls. Fine. Songbirds with similar calls are likely to be No. They are saying, I think you misunderstand what the question is asking. They say, I have, let's say, 2 different species. How will you know the relationship of these 2 different species? The relationship between species is always either closely related or distantly related. So again, you misunderstand. Okay? Next one, adaptive radiation. There is variation in ancestral population, and then they colonize different ecological niche. There's different changes in the leaf frequency in different things. Why you never talk about the natural selection? This is still a natural selection. I say macro, they always have to talk about natural selection one way or another. So you never tell me how there's different selection pressure in different niches. And then again, different alleles are passed down. And then you have different changes in the leaf frequency over time. So you're missing a lot of that. Again, you say there's different allele frequency changes in different populations. I can still give you the credit there. There's no gene flow. How come there's no gene flow? You never tell me why there's no gene flow. So this is the quotation of data you need. What kind of isolation they have that result in no gene flow? Is it geographical or reproductive? Reproductive, because again, they are different in eye color, plumage color, or male song variation. So you never tell me why. You cannot just say no gene flow. Okay? So you write down different changes in allele frequency, and then you never tell me you cannot just write no gene flow. I cannot accept. Okay? So you must say how can accumulate greater genetic difference. Fine, due to your natural selection. Fine, I can accept. So species no longer able to interbreed to form viable fertile offsprings. This is the behavioral isolation that you have to write in front that explain why no gene flow. Okay, fine, because you have behavioral isolation such as the different eye color. Fine, I can give you the credit here. Then with that, only then if you have that, then right in front of the no gene flow, then I can give you the credit. Okay, you say St. Patrick's speciation occur, I can still give you a maximum 5 marks. When it comes class, answer sheet is actually behind you. But I just want to go through, based on this, you already can tell, okay? I already show you how you can recognize, okay, what the type of questions that you're testing for evolution. For question 2A, they already mentioned what is the mechanism. Key phrase is called isolating mechanisms. There are only 5. They are your behavioral, temporal, mechanical, both post-zygotic or geographical. So you always ask yourself which of the 5. Usually, you cannot just write reproductive isolation mechanism. You need to specify what kind of reproductive isolation mechanism, if there is. Okay? Now, we already know the moment is geographical isolation, it can only be allopatric. But let's ask Chloe, because I never get the mark for you for this. We look at 2B. Do you see 2B here? They say it has been suggested that speciation is taking place. Explain how this process is occurring in the house mouse population. Can you tell me, is it micro or macro? Both. Why? What are the phrases that we see in the question that I know that I need to talk about both? Speciation already tells you form new species, so it's macro. But they also use the word how. Agree? And you look at the number of marks. Isn't it 5 marks? So another thing I mentioned, if you need to talk about both micro and macro, do you see how the key phrase is the same? But I already mentioned, yes, they always have the key phrase. But girls, don't just blindly vomit out the answer or key phrases. You must always write in the context that answers towards the question. You understand? Okay, so do you see in the answer sheet right now? Same thing, because this is what? Macro. I cannot just say variation due to random mutation, right? Highlight the word ancestral population. And then you must tell me how this ancestral population was scattered into different populations that evolve independently from each other. So again, same thing. You must tell me it's due to human migration and settlement. That's why, right, the population scattered into, in fact, 6 different valleys or 6 different populations. Okay? So, however, don't just tell me, oh, the steep mountain. Okay? So they are separating as physical barrier. It's because you already know it's geographical isolation. That's when the no gene flow comes about. I need to specify. When I say no gene flow, it can be due to what? Tell me the type of isolating mechanism. Don't just always write no gene flow, no gene flow. It can be your this reproductive mechanism or your this geographical mechanism. Specify. So in this case, it's the steep mountain, that's why there is no gene flow. Those who do the songbirds one is the reproductive mechanism, that's why no gene flow. So specify. And then again, different highlight, different selection pressures. Highlight selective advantage. Highlight changes in allele frequency. Do you see all these are all the generic keywords that you see for your this natural selection? But help me write, because this is macro. Results in different changes in allele frequency in different populations. For number 6, can you help me alter this slightly? Result in different changes in allele frequency in the different populations. Now you need to specify. For me, right, you must tell me there's different chromosome numbers because they clearly say how you have again different diploid numbers. And then do you highlight for me accumulation of sufficient genetic difference, or you can say accumulation of greater genetic difference. Isn't that what I mentioned? Unable to interbreed to form viable fertile offspring. Highlight that. Isn't that the key phrase? I'm just showing you this is one school. Later we do another school. Do you see all the key phrases come about again? So that's why I say these are the generic keywords. The beauty about evolution is there is always a lot of points and you just need to make sure you get the maximum mark. So write excessively. Next one for part C. They ask for likely outcome. A lot of students just tell me, okay, you have no longer able to interbreed. But I want to point out, examiners are never looking out for answers that are repetitive. You already know no longer able to interbreed should be a previous keyword already. It will not be repeating for this particular question. So this particular question is asking you what will happen to the offspring. They are saying likely outcome of individuals led. And then some of you tell me about the parents for what? They are saying the offspring themselves. So the idea is that offsprings are going to be infertile. But to me, if you tell me infertile alone, no marks. You must elaborate why. Why? Do you see the command word is called explain, class? I emphasize so many times. The moment you see explain, elaborate with effort. Unless this is a describe outline question. So you need to elaborate why stir around. So idea is that no pairing of homologous chromosome or there is absence of homologous chromosome, hence no viable gametes can be produced by the offspring. That's why they are infertile. And you cannot stop there. You must relate because they will have different chromosome number or diploid number that is given in the question. Okay, then I can give you the full credit here. Clear? Next one. Can we look at question 3A and 3C? 3A first. Keyword in the question. This is a suggest question, but keyword in the question is called calls for different species and the relationships. These are the two key phrases. That's why 2 marks. So again, a lot of students misunderstand what this is about. They are not asking like how the calls can help you identify the species. The question is asking you how come the calls can show the relationship between different species. That means I have 2 different species. How you show the relationship is always either how they are closely related or distantly related. So that's why, right, again, highlight the word species specific. Usually calls, if you want to identify, okay, as a trait, it must be species specific. Then you tell me, that's why the second mark is relationship. The greater the similarity in calls, the closer the relationship is between the 2 different species. Specify. Next one. You notice how this is part C? Never ignore the diagram, okay? Never ignore the paragraph. Please, they say with reference to, that means no matter what, you need to code 1 data at least. Even though it's diagram. Now, again, do you see the word adaptive radiation? Already tells you it's macro already, right? You look at the number of marks, you know that I also need to talk a bit about micro, but you need to tweak it to macro, right? Agree? But you notice how the moment, how come I know it's adaptive radiation? Besides the word adaptive radiation in the question, sometimes they can remove the word, right? But how do I recognize? The moment you see more than 2 species are formed, and in this case they said give rise to at least 7 different species, right? So I know this testing you adaptive. So what are some key phrases that you mentioned about adaptive? Can you highlight colonize different niches? highlight the word ancestral species. Because I know this is the adaptive radiation and talking about macroevolution. Do you see you still need to tell me about your micro? Myra, do you see now? Different locations or different niches have different selective pressures. You don't need to specify what is selective pressure, but you still need to tell me that have different advantageous traits that will be selected for. And that's why, again, differential reproductive success. You can also write out leading to differential reproductive success, such that different alleles are passed down, okay, to subsequent generation. And again, over time, there's different changes in allele frequency in different populations. You see we have here? But we know it's macro, right? That's why you also must tell me how come there's different, right? Specify the behavioral isolation. If you just tell me, okay, because they have no gene flow due to behavioral isolation, no marks. Because the question say with reference to. And this is the time that you have to code the data. They have different, they have no gene flow, okay, due to behavioral isolation, such that different population have different, okay, mating rituals, such as different male song variations, okay, or maybe different preference for eye or plumage colors. And then do you see the last one? Can you highlight accumulation of genetic different changes due to? Isn't that the same? It's acceptable, okay? And then same thing, no longer able to interbreed to form viable fertile offspring. Highlight that. Do you see exactly the same? That's what I say. These are all the key phrases. There's always going to be repetitive. The moment you can recognize it's micro or macro, please know what the keywords you have to input into your answers. Understand? Okay. Okay. Put this aside. I need to do classification. Classification, I'm going to summarize. I'm not going to do binomial nomenclature because you all should know what binomial nomenclature is. It's a scientific name. Okay, your genus and your species name. Usually when you write down in your answer sheet, you must always, what must you always do? Only underline all the time. Okay, come. For binomial nomenclature, I say already, I want to focus on taxonomy and phylogeny. There are the two different kind of classifications. There are two differences between these two types of classifications. Do you know what's the first type, one name? This one? When you classify your organisms,
Doesn't take into account.
so it may not take into account.
Evolutionary relationship.
Yes, evolutionary history, evolutionary relationship. Between the organisms. That means they may not consider whether they have or share a common ancestor. It's just similarities. This one always take into consideration. Okay, evolutionary relationships between species. Next one. What kind of classification system does this actually follow? Can you tell me, Xianfei? What kind of classification? You know the, at least can you tell me the classification? Is what? Sadara, King Philip came over for Masu is what? Please take note, you are expected in syllabus to know the sequential order when you classify the organism into groups. Okay? Kingdom, followed by phylum. Okay? King Philip came over for Granny's soup. You are expected to know, okay, three things. The sequential order for this. Please take note, those groups of organisms that has the largest similarity is the species group. Okay? We are also expected in syllabus to know, okay, what is going to be the humans, alright, kingdom, order, as well as this genus and species. So let's ask, can you know what's your kingdom? You are a human, so you are expected to know what's your kingdom. What's your kingdom? Sorry? Simi Homo sapien. Hello, Homo sapien is the Homo, which is the genus, and the sapien is your species name. But what am I missing? Yeah. What did I say for genus? And must always underline. Understand. Next one. Help her. What's your kingdom? Animalia. Animalia. And then again, we also need to know your order is what? Primates. Because again, I said you just need to know this. Primates are the ones with thumbs. Okay? Now, so for this taxonomy, you see it's all in what kind of classification system, Megan, that I mentioned? Hierarchical. Okay, classification system. It follows a hierarchy. Okay. C-H-I-C-A-L. Where's this one? You follow what? You classify organisms in a? Okay. Now, let's try. There are two types of phylogenetic trees. Cladograms and phylograms. Let's learn how to identify. And both of them have what we call clades. Now, first thing I want to mention is we need to learn cladograms. If I were to draw, this is a typical cladogram. There is no period of time located here. Sorry, can be a period of time, can be homologous. Phylograms, you can write horizontally, vertically, doesn't matter. But branch length matters. So let's try. Megan, usually this line can be time. But if it's not time, what does this usually represent? This main backbone usually represent? What does the main backbone represent? Whole. Yes. So again, what do I need? The main backbone usually represents your homologous structures, okay, that is shared from a ancestral species. If you were to flip under the classification system, do you see we have this? On your third column, I want you to focus on this example here, where you showcase certain homologous structures that we have from this lamprey, lancelet, tuna, and etc. So do you see the first one is like a vertebral column? That means the spinal cord. So all those lancelet, okay, do not have a vertebral column, they branch away. And then followed by the next one, we say jaws, okay, because you know so it means that the lamprey does not have jaws, but the lamprey has this what do you call that, vertebral column, and so on and so forth. So each line, okay, before the branching always represents a homologous structure that they have in common, and it must have been derived from an ancestral species. Okay, so take note about this. So again, my question is, your main background is a shared ancestral character, alright? What does this branch point actually represent? Branch point represents, again, the time. The closer the branch point is to the start of the main backbone, okay, the earlier at which this branching actually occurs. I repeat, okay, branch point closer to the start of the main backbone occur at an earlier time. Branch point closer to the end occurs at a later period of time. Okay? Cladograms, please take note, are fundamentally based on morphological data. Alright? Next one. Branch length. Does it actually matter? Like this one longer than this, does it actually matter, Chloe? No. So branch point, it matters, but branch length for cladograms are meaningless. Understand? The other one, phylograms. Notice how I have your this phylograms. Species 1, 2, 3, 4. In this species 1, 2, 3, 4, let's talk about this again. Branch point and branch length. In this case, there is no main backbone, but every time they have a branch point that I write in black. Does branch point mean anything, Megan? It means speciation has occurred. I formed a new species. Alright? So, it's a divergence of new species from a common ancestor, which is this. But does the branch length matters, Megan? For here? Just now here, branch length is meaningless. How about this? Branch length here shows what? No. Can you help me write down if it's not in your notes? Okay. Branch length show the number of differences in base sequence of DNA between descendants. Number of differences in base sequence of DNA underline, which arose due to mutations accumulated since divergence. I repeat, which arose due to mutations accumulated since divergence. So you start to realize that phylograms, the relationships are mainly based on molecular data. Right? Agree? So, before I go through, please take note now. Branch length matters. So if I were to have a branch length that is shortened, what does this actually represent, Myra? You see how here your branch length 2, 3, 4 end up at the same point, right? So how about 1? What does 1 actually represent? Because it's shortened. It is extinct. Please take note, the opposite of extinct is extant. Okay? Now, however, we are expected to learn clades, and clades are very common in phylograms. Let's ask up. First one, anything that's here is monophyletic. By definition, by the way, all those definition is in your notes already, second column. Monophyletic, what does that actually mean? Valentia? Monophyletic. What does monophyletic mean? You are expected in syllabus to know, monophyletic. You're this paraphyletic, polyphyletic. See in your notes there, quick. Because they are called clades. Wani, you will tell me the definition of clades. Can see?
A group containing only all the descendants of a particular ancestor.
All descendants of a particular ancestor. So if I want to draw monophyletic, I can draw these. These are all containing all the descendants of a particular ancestor. Okay? Okay. Paraphyletic means what, Xian Fei? They contain
Not all this.
so some but not all. Some but not all descendants from a common ancestor. Example, if I want to draw paraphyletic, this is paraphyletic. They do not contain species 1. Make sense? Okay? Please take note, it is inclusive of your this ancestor species as well. Alright? Next one. Polyphyletic means what, Chloe?
Some members do not share common ancestors.
There are some members, but they all do not share the same particular common ancestor. Example can be this. Some members, they do not share the common ancestor. This one missing, this one missing. Agree? This one also missing. So take note about that. Okay? Now, all these are clades, right? What's the definition of clades?
Come from the same lineage. No.
Help me write down the definition of clades if it's not there in your notes. A clade is a group consisting of a single common ancestor and all its descendants. I repeat, a group consisting of a single common ancestor and all its descendants, where members share similarities in DNA sequences. Where members share similarities in DNA sequences. Underline single common ancestor and all its descendants. So, Wani, with that, which one is actually a clade? Single common ancestor and all its descendants.
The poly.
Go ahead. Mount up. Okay, clear? Now, so you can see, right, the one that shows you relationship, okay, based on different species is actually which one? Phylograms. And this is fundamentally based on molecular data, right? I have not gone through this with any one of you all. We have nuclear data, DNA. So when we compare DNA, we always have nuclear and mitochondrial DNA. The question is, if I want to compare between mitochondrial DNA versus nuclear DNA, which one is actually more advantageous or more useful for evolutionary studies, Megan? Which is more useful for evolutionary studies? Mitochondrial DNA or nuclear DNA? Nuclear DNA, your head. Mitochondrial DNA. By the way, you asked me, right? Can you tell me, mitochondrial DNA, where do we get the mitochondrial DNA from? Mom or dad?
Mom.
Why? Just for context, this is maternally inherited. Why? 为什么啊? Oi. Question. Your mother's, the ovum, right? They will contribute every single cytoplasm, or is it the father's sperm can also contribute the cytoplasm? Because that is where mitochondria is located.
The mom.
The mom. Please take note. Only the mother's ovum, okay, will contribute the cytoplasm which contains the mitochondria. Father only contribute what? Xianfei? Oh. The nucleus, understand? So take note. Okay, this is just for context, but let's ask. Because I said mitochondrial DNA is more useful, right? We're going to learn how come. There are actually, again, two main reasons why mitochondrial DNA is more useful than nuclear DNA. First of all, mitochondrial DNA is almost like bacteria, right, Megan? It's what kind? How many copies? One. So if you have one copy, my question is, just like bacteria, do you think there's such thing as crossing over? So in other words, any difference that I see for mitochondria should not be due to sexual reproduction or meiosis or any other things, right? It's purely due to mutations. That's why it's very easy to study. You understand? So mitochondrial DNA is haploid, or you can say it's a single copy. Okay, so reason why is this useful? Because it does not undergo crossing over. like your nuclear DNA. I'm comparing with nuclear DNA. Okay, so when it doesn't undergo crossing over or even recombination processes, okay, like viruses, what does this mean? It means any differences that we see is purely due to mutation. Okay? So I can, and some more, every single organism has a mitochondrial lab. Right? Second reason. Usually mitochondrial DNA is used to study closely related species or distantly related species. Whenever we want to compare, let's ask Myra, can I ask, for closely related species, do you want to compare genes that are rapidly mutating or genes that are highly conserved? Closely related first. That means like chimps and humans. Do you want to investigate rapidly mutating genes or highly conserved genes? But the thing is, if you're highly conserved and you're so closely related, isn't that majority of genes is almost identical? Then how are we going to define? So that's why to compare closely related species, we are always comparing rapidly mutating genes like mitochondrial DNA. Whereas for distantly related species, humans and dogs, you cannot compare rapidly mutating genes, right? Because there will be so much differences that you will be mapos, right? Agree? So you compare highly conserving genes. Understand? So in this case, mitochondrial DNA helps to investigate, okay, relationship of your this closely related organisms because they mutate, okay, and they mutate in a high rate. And not only that, it's a clockwork kind of regularity. Is like you can estimate the time of even speciation. So two advantages from this. First one, can distinguish, okay, between closely related species. Which one is more closely related? So example, if you want to investigate chimpanzees and humans and orang-utans and humans, we can see which one has a closer relationship with humans, right? Another one is what? You can more accurately estimate time of speciation. Okay? So take note. Okay, now, so this is actually the molecular data. One thing I want to mention, they also like to test another thing, which is what is the advantage of your molecular data, right? There are 6 advantages. Let's see whether you know. 1, 2, 3, 4, 5. Just nice. Anana. First one. What's the first one? I want to hear the keyword for the advantage of molecular data. Myra? What's the phrase we always write side by side? You know what? First one, let's say, do you think that I actually can have any subjective judgments if I want to compare with molecular data, morphological data? No, right? Because every single organisms, if you are living organisms, you have DNA. DNA are what bases? What are the 4 bases? So, do you agree can be easily recognized? And one cannot confuse over another? What's the phrase we always say for advantages? You always write together side by side. Unambiguous and objective. Yeah, write down lah, so you remember. So again, use the phrase I want to hear is unambiguous and objective. Reason being, if you want to elaborate, because ATGC are easily recognized and cannot be confused with one another. Okay, easily recognized and cannot be confused with one another. Second one, if you want to write in another phrase, instead of being saying cannot confuse with one another, you can say not dependent on subjective judgments or observations. Okay, observations involving, let's say, qualitative differences in morphology. Okay, so take note about this. Unambiguous and objective is always the keyword. Second one is what? Can you tell me, Colentia? I can. Can I quantify it? And then put it into what form? Always must write together. Plus, minus, divide, times is what? The numerical or mathematical form for what kind of analysis? You want to know how come we can get 98%? All this is what kind of analysis? Statistical analysis. Number 2 is quantifiable into numerical form. Quantifiable into numerical form and open to statistical analysis. And open to statistical analysis. Okay? Next one. Can I actually find out the differences between each species accurately? Like for example, humans and chimpanzees, right? Because there's so little difference, right? But there's still so many things that we can compare. Why? What is the way we can compare the many points of comparison? Because it's all based on like for example, right, I want to compare cytochrome C, right? Okay, between the chimpanzees and humans. So how do I actually compare this cytochrome C? Based on your DNA, right? Yeah. So we can actually find differences based on position of the nucleotides. Idea is that we also can take note now. Molecular data, homologous regions of DNA, from different species provide many points of comparison. I repeat, homologous regions of DNA from different species provide many points of comparison as each nucleotide position is a point of comparison. Provides many points of comparison as each nucleotide as each nucleotide position is a point of comparison. Next one. Can I avoid convergent evolution with molecular data? Yes, because sometimes, right, they look actually similar, but they have very large genetic difference. What? Will I therefore avoid the pitfall which morphological data tend to actually sometimes misunderstand? Yes. So another one is that avoids pitfalls of convergent evolution. Avoids pitfalls of convergent evolution. Okay, there's another one. Fifth one. Chloe, can you tell me, what do we use? Notice how we use molecular data to establish just now all the trees, right? What tree is that? What phylogenetic tree is dependent on molecular data? Philo? We only have 2 types of phylogenetic tree. What are they? Cladograms and phylograms. Which one do you actually consider molecular data? So isn't it based on this molecular data, which will help you trace evolutionary relationships? So another one is molecular data can be used to trace evolutionary relationships of species, especially if they are so different despite little morphological homology. I repeat, can trace evolutionary relationships of species, especially if they are so different despite little morphological homology. Okay, let me give you another example. For example, maybe I want to find what's the relationship between plants and bacteria. It's very hard to use morphological evidence or data to establish the relationships, right? But I can use the genes, okay, to establish. Clear? Last one you also can write is another one. That means you want to draw a phylogenetic tree as well. Number 6 can be used to assess phylogenetic relationships that cannot be measured by comparative anatomy. I repeat, establish phylogenetic relationships that cannot be measured by comparative anatomy or fossil data. Okay? You can also even study deceased organisms. Alright? Clear? Okay, come, let's try. You see we have again a few questions. Each of you will do your respective questions. Then I'll just give you about 7 minutes. Alright? Same group, okay, Xian Fei, Megan and Kalantia. You see we have question 1A and 1B. Okay? 1A and 1B. And then and 1C, okay, which is the entire question 1. Each of you do 1A, you do 1B, you do 1C. Okay? Next one, do you see we have here, which is actually question 3B. Wait, hold on. I repeat, 2D, 2E, and 3B. So, again, here, I want you to do 2D and 2E. Alright, Myra, you help me with 3B. Alright, and then you will actually help me with same thing, 3B as well. Okay?
So for the last one is establish phylogenetic relationship that can be determined by morphological data and
Establish phylogenetic relationships, okay, that cannot be measured by anatomy. That means comparative anatomy or fossil data. Okay? Okay. Anyway, I'll give you about 5 minutes to do your respective questions so you can go through class. Oh, I forgot there's one more. Just on the isolation mechanism, I forgot there's such thing as physiological isolation. Physiological isolation is when you have different chromosome number. Okay? Physiological isolation is the next one. Sorry, I forgot about that. Just remember for now. Okay, I'm gonna go through very soon. Give you another 2 more minutes. Yeah, don't need to write so much, Xian Fei. They are asking just straight away what does the length show you about the evolution of that particular species. Do you see it's shortened? So all you need to tell me is that the species is yeah lah, that's all. Okay, 1 mark only, always tell yourself. 1 mark, 2 marks, straight away give me very straightforward answers. Understand? Hopefully you know higher the percentage of this temperature, right? That means what? You need to break more bonds, right?
Yeah.
That means is it more hybridization or less hybridization?
More hybridized.
Yes, because there's more complementary base pairing, right? Which shows again they are more homologous, right, to each other.
Yeah.
Okay. You hear what I say, Myra? Okay. Sorry, because of time I have to go through. Can I go through? Okay, let's start off with question 1A. So they ask you to distinguish the classification for phylogeny. Hopefully you know this classification is what? It's a type of taxonomy, cladograms. So cladograms and all this kind of classification, please take note, it's also like taxonomy. Okay, so what is it going to be? Because do you see the homologous one? So can you tell me, distinguish these differences, right? Jennifer, what is the first difference that you have?
Evolutionary relationship.
Can you tell me what you write?
Phylogeny is determined based on evolutionary relationship, but classification is based off like morphology.
No, remember the differences I have? So the way you write, right, is organization of species. One does not, that means classification may not take into account consideration of evolutionary relationships. What the other one? Phylograms have. Okay? So take note, or you can say one is a hierarchical classification system. That means the kingdom, phylum, class, order, family, genus, and species. The other one is based on a phylogenetic tree that is dependent on evolutionary relationships. Please start this question so you can use the answers behind as a studying material. Class, do you see this homologous, this main backbone? This Tacitus, right, is actually a shortened one, right? It doesn't end off to the same end point, right? So what does this showcase about this species? Xianfei? The species is? For part 2. Species is? What does the length of the line represents the evolution of the Tacitus?
The species is extinct.
Yes, as long as you tell me the species is extinct, I will give you 1 mark already. Next one. Can you tell me, Xianfei, based on this diagram, we have the common bottlenose dolphins and the Buranan dolphins. Are they a micro or macro evolution event? I show you, right? There are 2 things that you look up for as evidence to see whether micro or macro, right? So is it micro or macro? macro. What are always the two evidence we have to come up for? This results in formation of, are they different species? Yes, right? The moment you see two or more species, there's already macro. Micro is within the same species. Another thing is, what did I say? The changes occur over how many years?
More than 1 million.
You don't write more than 1 million, it's millions of years. So it's a long period of time. So you need to give me both, then I can give you the credit. Next one. We have the fossils here. So the fossils actually provide evidence, and we have an impression of this Ambulocetus. So now they say, explain how the morphological evidence supports theory of evolution. Answer sheet is not very good. So keyword in the question, do you see theory of evolution? Agree? So what must we always include if you want to explain theory of evolution? Megan? So I want to see the word descent with modification. You see in the answer sheet, they have a homologous structures. It's also the keyword. But again, can you tell me? They all share the same basic plan in structure. That means they all have head, body, and tail, right? Agree? So all you need to tell me is that because they are same basic structure, and this must be derived from a common ancestor. Okay? So I want to see the word descent or descent from a common ancestor. And then the other one, you must tell me how it's modified, right? Based on natural selection. Highlight the word different selection pressures. Okay? Idea is that not only you must tell me face different selection pressures, okay, to serve for different function. Can you help me write after the selection pressure? You write the word for different functions. Next one. Keyword in the question is what? Transition fossil. Structural features. Do you notice how it's 2 marks? So first mark is showing me how it's adapted to land, and next mark is telling me how it's adapted to water, right? So what feature did you see there, Colantia? I don't want an answer sheet, I want the diagram because I have more than whatever answers they have there. So how is it adapted for land? Can you tell me? What does this diagram have? They have?
I said they have 4 limbs.
They have 4 limbs, okay? F-O-U-R, okay? 4 limbs, okay? For walking or locomotion on land, alright? I can accept, alright? Another thing, for adaptation of water, what do you see there? Look at the feet.
Wet.
So they are wet feet, alright? To propel in water, or move in water, or swim in water. Another one, what do you see? Do you see I have a streamlined body for swimming? If you write streamlined body, I also can accept. Another one, I also can accept. Notice how, okay? The eyes is actually, we have a long snout and eyes near the top of the head. Purpose is for what? For ambushing prey in water. Almost like a crocodile or an alligator. So again, for adaptation of water, you can say wet feet for swimming. You can say streamlined body for swimming. You can say this long snout and eyes near the top of the head, alright, for ambushing prey in water. Anything that sounds logical out of this out of this tree, name me one, I'll give you the full credit. Next one. We have here, which is actually question 2D and E. So in question 2D, they say state one feature of the cells in the kingdom Animalia that distinguish them from cells of other multicellular eukaryotes, which are plants, right? So if you want to compare with the kingdom Plantae, how is Animalia going to differ? That means they're asking, what's the difference between animal cells and plant cells? Oh, sure, no cell wall. I can accept no large central vacuole. I also can accept presence of centrioles. So always try to write in an active tone. Understand? Next one, they say why amino acid sequence of protein can reveal useful evolutionary relationships. Because if I change amino acid means that can tell you your evolutionary relationships, Chloe? Why? Why? Amino acid is encoded by what? Which are? DNA sequences. So if you change your amino acid means what is change? DNA sequences is change. So it means mutation must have occurred, right? So in other words, if I were to have a larger difference in amino acid sequence, what does it tell you about the evolutionary relationship? Because the keyword in the question is how you review evolutionary data. Evolutionary data is always relationship, right? So the larger the difference in amino acid sequence, what does it mean? The more this, huh? How is the relationship going to be like? I told you the relationship is always got to do with closely related or distantly related, right? So is it closely or distantly related? More distantly related. You understand? Okay? So you need to reflect back to your DNA because it's fundamentally based on your genes or your DNA sequence. Next one, can I ask? In this case, notice how we have hybridization, right? Myra, can you tell me? Notice how if I say, keyword in the question, temperature at which separation occur can use to determine percentage of DNA hybridization. Why? What's the relationship between temperature and percentage of hybridization? Higher?
Percentage of hybridization require higher temperature.
To separate. Agree? But this is explained, right? I need to elaborate with effort, right? So why?
So higher temperature
It's needed for what?
is needed to break more bonds.
What kind of bonds?
More hydrogen bonds
Between?
the nucleotide bases of the DNA.
Between complementary bases. Okay? So remember, they are using a unique DNA hybrid, and then the more they can attach, that means the more hydrogen bonds they actually form. That's why it requires more energy by temperature, by higher temperature to break those bonds. Okay, Myra, can you tell me which are the two that are the closely related ones? There are only 3 species, so which are the two that is most closely related? You can draw vertically or horizontally, I don't care. So which are the two closely related, Myra? Based on the percentage?
The first one.
Which is spinous, okay, as well as the cardulis, right? Then, show me, you must reach at the same point. If your lines did not end off at the same point, you will be penalized. This is the SC. What must I always do, Myra? Because I'm writing down the genus and species name. Every single one you must underline. If you never underline, you will be penalized. Understand? Okay, so criteria, end off at the same point. Number 2, underline the genus and species. So with that, I end off here today. So I repeat, there are 4 types of questions they like to test for your this evolution. Your theories of evolution, please know what are the keywords relevant. Microevolution, macroevolution, I showcase how you can identify. 4th one, classification. The classification wise, they either ask you how is the molecular data can be used to establish relationships, how you can draw your phylogenetic tree, and the most common one, advantages of molecular data. Please never lose marks for your this evolution. It's usually based on a template or standard keywords.